Early on in this course, we learned that genes are able to express themselves in the form of proteins. So we have genetic material in the form of DNA in each cell of our body. Then there is production of RNA called the messenger RNA from DNA in the process of transcription. From RNA, we can synthesize proteins, and that information in the DNA is translated into proteins. Now the genes are expressed as proteins. So this is the central dogma of life. That is, the DNA sends its information to RNA and RNA sends its information to protein. So all the characteristics in your body are manifested through proteins, because proteins form hormones, they form structure, they form function, color of your skin, color of your hair, color of your eyes, height, weight, everything is programmed through proteins. So the cell function, development, and all the responses to its environment are determined through what genes a cell is expressing. Although each cell in your body has all the same genes, still cells perform specific function. For example, muscle cells are involved in contraction, eye cells are involved in visualizing light and color, and cells in your tongue are involved in perceiving taste. So by definition, gene regulation is the mechanism that controls timing, location, and the amount of genes expressed. Why is it important? So that each cell can perform its own defined function, conserve the energy so that resources are not continuously used, and it allows for cell differentiation, which means the muscle cell would be different from eye cell and the eye cell would be different from the cells in the tongue. The level of regulation are at the level of transcription, after transcription, at the level of translation, or post-translational, that is after translation. Now let's look at how genes are regulated in the prokaryotes at transcriptional level. Let's say these are 4 different genes, that is a segment of DNA in the chromosome, and they'll produce RNA, and RNA will end up making 4 different proteins for 4 different genes. But how will these genes know that they have to start expressing themselves? That means, how would they know that they have to send a messenger RNA and then lead to the formation of specific proteins? So this system is called the operon system, in which you have some regulatory genes and some operator genes which come together and coordinate the expression of different genes. So the key players of this system are RNA polymerase, promoters, operators, repressors, and inducers. These operon systems have 2 kinds of regulations. A negative regulation, in which the repressor prevents the transcription of a gene. So then you'll have to remove the repressor for the gene to be able to translate. The other mechanism is positive regulation, in which the activator is continuously present, leading to the expression of gene continuously. So those genes are always required to produce proteins, and they are called the housekeeping genes. But how would you stop those genes from expressing themselves? You will have to inactivate their activators. So now we look at the example of one of the operon systems called the lac operon, which is present in E. coli, a bacteria. So this lac operon system controls the breakdown of lactose. We know that lactose is a sugar that gets broken down into glucose and galactose. There are 3 genes required to produce the enzymes required for the breakdown or metabolism of lactose. These genes are LAC-Z, LAC-Y, and LAC-A gene. But this system is regulated by a promoter and an operator. And there are some genes called the LAC-I genes that produce a repressor. So most of the time a repressor is continuously produced, which sits on the operator and prevents the promoter from promoting the transcription of these three genes. Whenever you want these genes to express themselves, you will have to remove these repressors so that the promoter can now ask these genes to express themselves. So how does it work? When there is no lactose, that means you don't require these three enzymes. That time the repressor is sitting on the operator and blocks transcription. When lactose is present, the lactose binds with the repressor and changes the conformation of this repressor. Then repressor can no longer block the operator and the promoter is able to send a signal for the transcription of these three important enzymes. To summarize, when there is no lactose, the operator is blocked by the repressor. At that time, RNA polymerase cannot promote the transcription of these three important enzymes required for breakdown of lactose. You don't need them because there is no lactose, so you don't need these enzymes anyway. But when there is lactose, the lactose will block the repressor from binding with the operator. Then this path is open for the RNA polymerase to induce the transcription of these three enzymes. Why would you need that? Because when lactose is present, you want these enzymes to be present so that the lactose can be digested. A different kind of mechanism is seen in tryptophan operon. Tryptophan operon also operates in E. coli, and in this we'll study the genes related to the synthesis of tryptophan, which is an amino acid. So you always require tryptophan. It's not a situation where you would suddenly require tryptophan. The E. coli continuously requires the synthesis of tryptophan. Tryptophan production is regulated by these 5 genes. Other than that, there is promoter, operator, and a leader sequence, and there is a repressor. Now let's look at the mechanism. Continuously, these 5 genes will produce their protein, and these proteins are involved in the synthesis of tryptophan, which is continuously required by the bacteria. But when there is suddenly a high concentration of tryptophan, it binds with the repressor, and then the repressor can bind with the operator and block transcription. Unlike what you saw in lac operon. In lac operon, the genes were not continuously synthesized. Only when lactose was present, lactose would remove the repressor from the operator so that the genes could express. Here in tryptophan operon, genes are continuously expressed. In this situation, tryptophan binds with the repressor, and then repressor binds with the operator and blocks the transcription process. So when the tryptophan concentration is low, at that time the repressor is inactive because it only gets activated by the presence of tryptophan or binding with tryptophan. So when tryptophan is low, it's not binding with repressor anymore, it becomes inactive, and that time it gets removed from the operator gene and transcription can then proceed again. So let's summarize this mechanism again. In the presence of tryptophan, the repressor is active, and that time it can bind with the operator and block RNA polymerase from synthesizing RNA or block the transcription process. But when there is no tryptophan or low tryptophan, the repressor can then not bind with the operator, and it will lead to continuous formation of the mRNA from these genes. That means the transcription process continues in the absence of tryptophan. Whereas in lac operon, the system would not operate in the absence of lactose. The system would get activated only in the presence of lactose. So the two operon systems that you just saw, the lac operon and tryptophan operon, are much simpler to understand, and they are present in E. coli, and E. coli is a prokaryote. In eukaryotes that have a much more complex nuclear structure, the transcription system is not that straightforward. So there are no straightforward operon systems in eukaryotes. The regulation is much more complex. Even the DNA present in the nucleus in the form of chromatin has a very complex structure. It can be very much coiled or supercoiled, so it's not very easy to regulate those kind of genes. The promoter present in eukaryote is a TATA box, which is thymine, adenine box. And eukaryotic system has enhancers which bind activators far from the gene. The transcription factors have DNA binding and activation domain. The enhancer is much far away from promoter and the gene they are supposed to transcribe. Then a transcription factor comes in. The transcription factor binds with the enhancer, and this creates a hairpin loop, and they end up activating the promoter. Then RNA polymerase sits at the promoter and leads to the transcription of the gene which is present next to the promoter. So in eukaryotes, if you know that DNA is wrapped around histone proteins in the chromosomes. So the DNA is coiled like this around the histone proteins. So the histone proteins, if they are acetylated, that leads to the activation or enhancement of transcription process. But if the DNA is methylated, there is a methyl group attached to the DNA, then it leads to the silencing of transcription. So let's look at the comparison of eukaryotic and prokaryotic gene regulation. In prokaryote, there are operons. So multiple genes are expressed under the regulation of same set of operators and repressors. But in eukaryotes, individual genes are regulated individually. In prokaryotes, the chromatin structure does not play a very important role, but in the eukaryotic system, how the DNA is coiled in the chromatin plays a very important role. Sometimes the coiling is so tight that the genes are not easily transcribable. The major elements of regulatory system in prokaryotes are promoters and operators. But in eukaryotes, of course, there's going to be a promoter because the RNA polymerase comes and sits at the promoter. Other than that, you have enhancers and silencers. In prokaryotes, there are very few transcription factors, but in eukaryotes, the transcription factors are numerous and their interactions are much more complex. So gene regulation has several applications. For example, in biotechnology, you can make several genes to express, which leads to the production of the proteins that are required for their industrial purposes. Similarly, when combating certain diseases like cancer or genetic disorder, you can make some gene repress or be more enhanced depending on the requirements of the protein that is supposed to deal with the disorder. And in evolution, of course, through the process of evolution, several genes have adapted to be more expressed or less expressed depending on how the organism needs to adapt or evolve in accordance with its environmental factors. So to conclude, gene regulation is very crucial for all kinds of cells. We just saw an overview of operon system, which was lac operon and tryptophan operon in case of prokaryotic system. And we also realized that eukaryotic regulation is much more complex because of the chromatin structure and presence of transcription factors, and also that transcription factors play a very important role in the expression and regulation of gene in eukaryotic system. The study of gene regulation helps in advancements in the field of medicinal research or agriculture and biotechnology.